How is messenger RNA read? Actually, the genetic code. It's a universal genetic code used by all living organisms, and as you know, it works in 3 nucleotide units called codons. Each codons codes for single amino acids. There is as well 1 start codon and 3 stop codons. You can see from the table. As you can see from the table, you can see as well that 1 amino acid may be coded by more than 1 codon. The messenger RNA have possible 64 possible codons. It's actually the combination of 4 times 4 times 4. This is a universal code that specifies amino acid. 61 codons for amino acid and 3 codons for the stop codons. Only 2, methionine and tryptophan, have a single codon, while leucine, serine, and arginine have 6 different codons. The synthesis of proteins and prokaryotes must begin with methionine. The initiator codon is AUG. Some other codons like GUG and CUG are rarely or sometimes used as initiator codon in some eukaryotes or few bacterial messenger RNA. What is reading frame? Actually, its reading frame is a sequence of codons that start from a specific start codon to stop codon. Messenger RNA can theoretically be transcribed into 3 different frames, but most messenger RNA can be read only in 1 frame because the stop codon is encountered in other 2 possible frames and terminate the translation before the functional protein is produced. To have a more illustration, this is frame 1 of translation. So messenger RNA is translated into frame codon by codon by codon. In frame 2, there is a shift of the first nucleotide is ignored in the process of translation. So you'll have a shifting. That's why it's called frame 2. So the codes have been changed and different protein, different polypeptide is formed. As well as in frame 3, when the first 2 nucleotides of the messenger RNA are ignored and another protein is formed. Normally, the messenger RNA is read on only 1 frame, and normally it's a frame 1. If you look closely here to the table, so this first column represents the first position while reading in the process of translation, while this second position of the triple code represent these 4 nucleotides, and this is the third position. So this is the 3 position. To have an example, if you have a DNA that have this sequence, and you want translated, you want to transcribe it into messenger RNA, this messenger RNA will be transcribed in this way. Keep in mind that we always read the nucleic acid from the direction 5-3-. So actually, TAA can be transcribed into AUU, but we needed to write it in the correct orientation from 5- to 3-. Okay, again, TAA, the first codon in the DNA, TAA is transcribed into AUU. And this is the 3' end. Okay, so normally we write the nucleic acid from the 5' ends. If you want to translate this, to make it, why you normally write it in the 5' end? Because it's easier to understand translation. So you have the first codon now is GCU, and this gives alanine amino acid, and you can see this alanine. And if you try to exercise on the other 2 codon, you'll find that they are translated into cysteine and leucine. The levels of gene expression. So the gene regulation is on different levels. It's not only on the level of the nucleus or the level of cytoplasm or the level of the translation. You have the first level, the regulation of gene expression, while the DNA got unpackaging and due to the involving DNA demethylation and histone acetylation, regulate if the gene is available for transcription, and this is the first level of regulation. While the second level is the transcription regulation. This means to transcribe or not transcribing the gene. The third level, the RNA processing, which I've seen it consists of 3 different processes, the capping and the polyadenine tail and the splicing of entrons and removal of entrons. the next level is the transport of the cytoplasm. It's a mode of regulation of gene expression as well. The next level is translational regulation and degradation of messenger RNA, while this is followed after the protein is translated into post-translational regulation, either by chemical cleavage, modification, transport to cellular destination, where the function of the protein is active. After the protein have been fully formed and active and done its function, it must be degraded for recyclation of the amino acids. If we continue of the level of gene regulation, we will find another category of RNA, which are non-coding RNAs, which its major type is microRNAs. Recently, microRNAs have been a lot of microRNAs have been discovered, and they found that these non-codings, nucleic acids, can silence genes, reveals another level of gene expression regulation. You can see that the microRNAs are transcribed from the DNA as a pre-messenger RNA, microRNA. Under the effect of DROSHA enzyme, it will convert it into pre-microRNA. There is some processing DROSHA inside the nucleus. Following the export of this pre-microRNA into the cytoplasm, under the effect of Dicer, there is a cleavage and the microRNA is formed. So let's try to explain in more details. So the microRNA genes transcribe a primary transcript called the pre-microRNA that folds into hairpin loop similar to the folding pattern of a transfer RNA molecules. This enzyme Dicer cleaves this pre-microRNA into single-stranded microRNA that are around 21 to 22 nucleotides. The microRNA bind to a cluster of proteins to form a ribonucleoprotein complex. This microRNA complex can bind to messenger RNA by complementary base pair, and this microRNA either bind and silence gene expression by blocking translation or by targeting the bound messenger RNA for degradation by enzyme in the cells. So it's actually another mode of regulation at the level of translation. The next subtopic is mutation, causes and consequences. Of course, mutation is a change in nucleotide sequence of DNA. It's a major cause of genetic diversity, can also be harmful. Types of mutation either point, silent, missense, nonsense, frame shift mutation. This is inherited through gametes or acquired genomes of the somatic cells. Human genomes are approximately 99.9% identical. There is only 0.1% difference between individuals, roughly 3 million difference between different individuals. Most mutations have no obvious effects. Other mutations strongly influence cell function, behavior, susceptibility to genetic diseases. Gene mutations can cause changes in protein structure, function, synthesis of non-functional proteins, or no protein synthesis can lead to a change or loss of trait or a phenotype. So biotechnology is being used to detect and correct mutations. If we look too closely, this is one of the important slides. If you look closely to the types of mutation, you'll see, if you assume that this is a normal wild type genes, have two double-strand DNA, a single-stranded DNA have been transcribed into messenger RNA. Okay, so we have a messenger RNA that's been translated into a polypeptide formed of 4 amino acids: methionine, lysine, phenylalanine, and glycine, followed by a stop codon, so there is no amino acids. And this is the normal behavior of gene transcribed into messenger RNAs and followed by translation. There are generally two types of mutation, either base pair substitution or base pair insertion or deletion. You will see that there is a silent mutation in which the nucleotide A is mutated from the original, because originally it was G if you look to the wild type. So this gives another nucleotide when transcribed that doesn't affect the amino acid sequence of the translated protein. That's why it's called silence, no effect on the amino acids. The other type is missense change codon. So you will see that T have been changed, leading to the change of the amino acid that has been inserted in the polypeptide. So that one is a different proteins. The other type is nonsense mutation, creates a stop codon. So the mutation in the DNA leads to formation of codons that lead to a stop codon. That means the protein formed is truncated only of methionine. Okay. The other category of mutations, the frame shift, means the frame shift. That means that you will see that the T nucleotide have been lost. So the codon start to read in the absence of T after the transcription. So two new amino acids have been inserted, which results in a protein or polypeptide that totally different from the wild type. While in frame shift causing immediate nonsense, that's mean insertion of the nucleotide A leads to formation of a stop codon. That means formation of a truncated version of the protein, as well as insertion of deletion of 3 nucleotide will affect the framing of the translation. So the reading frame will be distorted. So another another amino acid will be inserted because of the disappearance or the addition of 3 nucleotides. The last subtopic in this lecture is revealing the epigenome. So epigenome refers to the modification in chromatin structure which do not involve mutation DNA sequences. So there is a change in chromatin structure and no mutation is involved. Some epigenetic modifications are inherited, others vary among generations. Some these of the modification can be reversible, while others are long-lasting and irreversible. Epigenetic changes can differ between cell types in the body and in normal and diseased tissues. Diet and environmental conditions can influence the epigenome. It is now known that the epigenetics play a major role in clarifying how patterns of gene expression can vary during embryonic development. So you can see here that the chromatin involved in epigenetics. You can see that DNA methylation, histone modification, remodeling complexes, histone variants, non-coding RNAs. All of them control when a gene transcribed and how much of messenger RNA are transcribed, when it's switched on, when it's switched off. So it's all types of modification that have a level that can regulate gene expression without mutation, or can just change the protein without the protein pattern without mutation. And in next lecture we'll deal with recombinant DNA technology and genome. Hello, we'll continue our lecture about biotechnology with the fourth topic, recombinant DNA technology and genomics. We'll deal with 5 topics. Generally, the applications of recombinant technology is widely used in biotechnology, medicine, and research. It's used to identify, map, and sequence genes and to determine their function. Recombinant DNA is used to produce recombinant human insulin, for example, growth hormone, for example, insulin-resistant crops. Bacteriophage are viruses that infect bacterial cells. In 1970, gene cloning became a reality. What is a clone? A clone is a molecule or a cell or an organism that was produced from another single entity. It's made possible by the discovery of restriction enzymes, which are DNA cutting enzymes. The plasmid DNA vector is a self-form of self-replicating DNA generally present in bacteria, can be manipulated to carry and clone other pieces of DNA. What is restriction enzymes? Restriction enzymes is primarily found in bacteria. They use these for defense against viral infections because they lack immune system. These enzymes are endonucleases and cut DNA by cleaving the phosphodiester bond in DNA strand. They bind and recognize cut DNA with specific sequences called the restriction sites. Each restriction site is palindromic. What's meaning of palindromic? Means that reads same from forward and backward on opposite strands of DNA. Remember the word madam, you can read it palindromic, from right or left is the same. These enzymes cut DNA to create DNA fragment either with overhanging single strands ends called sticky or cohesive ends, or with double-stranded ends, it's called blunt ends. The advantage of this enzyme that can produce sticky ends, it is preferred for cloning because DNA fragment with sticky end can be easily joined together because they base pair with each other. Why don't restriction enzymes digest its own bacterial DNA? Actually, if we look to this diagram, we will see that DNA restriction enzyme, which is called EcoR1, can produce, after recognizing the restriction site, can reproduce the unmethylated form of DNA and decleave it into two cohesive ends. While in bacteria, this restriction site is methylated. You can see it is methylated. So once this restriction site is methylated, the EcoR1 methylase, the EcoR1 enzyme cannot recognize the restriction site. And that's how the bacteria protect its own DNA from being cut with the enzymes. This is like common restriction enzymes used. You don't need to memorize this figure. You can see there is three types of restriction sites which create blunt ends. In this figure, you can see four types of restriction enzyme: EcoR1, HEND3, BAMH1, and TAC1. They create cohesive ends or sticky ends. Would EcoR1 cut the following sequence? Explain your answer. Just check if EcoR1 can cut this sequence. Do it as an exercise. You can go for the 7 restriction enzyme, 4 here, for the EcoR1 restriction site, and compare and see if it will cut this sequence or not. Now, how to find the right restriction enzyme? So if you have a DNA in hand, how you find the right restriction enzyme for the right DNA sequence that you have? Using an online software, it's called NepCutter version 2. This is the website. This is the link for the website. You will have this kind of page. You have local sequence. So if you have the file already saved on your computer, and if you have the number, and instead of saving just the ID, if you don't have a number or the file and just have the sequence, okay, you need to choose if it's a plasmid vector or a linear DNA. So you insert, if you have just the sequence, just insert the DNA sequence here, and once you insert, you can choose if it's linear or circle, and then submit. Once you submit, here is the sequence, and you submit. Once you submit, you can see the sequence have been shown with all the restriction enzymes that can cut, and the location of the restriction cut. So you can see this small sequence of DNA can be cut by around like 1, 2, 3, 4, 5 different restriction enzymes. EcoR1, of course, you can see that it can cut this this DNA sequence. Here is the key for the figure. You can exercise doing the same with any sequence of your choice on the online platform. What is plasmid DNA? Small circular pieces of DNA found primarily in bacteria. They are around 1 to 4 kilo base pair, are considered extra chromosomal DNA because they are in the cytoplasm in addition to the bacterial chromosomes. They can replicate independent of the chromosome and can be used as vector. So you can accept pieces of DNA, carry and replicate other pieces of DNA. So if you can see the general plan of any plasmid vector, it consists of 3 regions. The ORI, which is the origin of replication. Let's start with the multiple cloning site, MCS, or the polylinker. It's a sequence of DNA that have all the restriction sites for these enzymes. And this is the region where the gene you want to insert into the plasmid is inserted here. So how to create a recombinant DNA? First, you have the DNA, the human DNA. The human DNA, if we assume that it will be cut by EcoR1, so it will produce a cohesive end like this, 2 cohesive ends. So you get this cohesive end, and you insert this cohesive end to the plasmid, which is have been cut by the same restriction enzyme used for the cutting the human DNA. So here you can see that the plasmid that have been cut by the same restriction enzyme, in this example it's EcoR1, have been ligated with the enzyme, with the piece of DNA that have been cut to form a recombinant DNA. It's like a straightforward experiment, doesn't takes like 1 hour in 37 degrees, needs the enzyme ligase. So finally, the recombinant DNA is formed by from insertion of a piece of DNA. The most important thing here that the both of the human DNA and the plasmid used for insertion must be cut by the same restriction enzyme to have the same cohesive end to facilitate the ligation. In this figure, you can see that we plate on LB agar. The LB agar contain ampicillin, then ampicillin sensitive E. coli cell in log phase of growth is transformed with the use of calcium chloride. Then we add the cells, incubate the cells with the cells are heat shocked at 40 T number 3. Heat shock at 42 degrees centigrade. This bacteria is called recombinant bacteria, will take up the plasmid because at 42 degree there is pores arise in the wall of the bacteria and take up plasmids, the recombinant one. Then we plate this plate these bacteria on the LB agar which have ampicillin and incubate overnight at 37 degree. The colonies, only colonies number 6, only colonies of E. coli that have been transformed by the ampicillin resistant gene will grow. Other one which will not, which didn't took up the plasmid will die. So to see the result of the transformation process, we took this bacteria and plate on no ampicillin in gross media and plate in ampicillin in gross media. You can see that the plasmid that you can see that in control 1 and control 2, no bacteria arise no bacteria have been grown on the plate with the ampicillin because there is no ampicillin resistant plasmid is added. Okay, so in this case, if ampicillin resistant is added, you can see that some few bacteria are grown on the ampicillin in the gross media. That means this kind of bacteria succeeded in taking up the recombinant plasmid. So let's place the stages of transformation of order. You can see that this is like a shoveled transformation steps, and if you look closely, it's A, B, C, D. You need to put in this order, and of course this is the answer. You can revision that. The first step is C, then followed by B, D, E, A. So continue our talk about selection of recombinant bacteria after transformation. There is an easy way to select using a concept called the blue-white selection. DNA is cloned into the restriction site. DNA is cloned. This means the gene you want to insert inside the plasmid. So DNA is cloned into the restriction site in the LAG-Z gene. When it is interrupted by an inserted gene, the LAG-Z gene cannot produce functional beta-gal. When X-gal, which is like artificial lactose, is added to the plate, if function LAG-Z is present, there is a blue colony. The colony of the bacteria will be blue. If non-functional LAG-Z, that means the LAG-Z sequence of DNA have been interrupted by insertion of a gene. A white colony will be produced. That's a genetically identical bacterial cell, each contain copying of recombinant plasmid. To summarize that, this means if I have a recombinant plasmid and if I have a bacteria and the bacteria give me white color, that means these bacteria have taken up the recombinant plasmid. If give me a blue color, that means these bacteria didn't take up the recombinant plasmid. Okay. Assume you use the plasmid that contain the LAG-Z gene in the restriction enzyme site. The plasmid has an antibiotic resistant genes. Following transformation, you grow up the cells on agar plate containing the antibiotic. And you can see here is your results. You can see this is like 3 white and 3 blue. Of course, the 3 white will taken up the plasmid, while the blue will haven't taken up the plasmids. You can, again, for the blue-white selection, so you can see here, in one, you isolate the plasmid, which you have a bacterial chromosome and the plasmid. You need to cut both DNA sequence with the same restriction enzyme, and this plasmid will have now the LAG-Z gene, you can see, and the ampicillin resistant gene, AMP-R, and this plasmid have an origin of replication. It's the region which you control the replication of the own replication of the plasmid. So now these plasmid have a LAGZ gene. This LAGZ gene contain a number of restriction sites for restriction enzyme. You can see 5 different restriction enzyme: EcoR1, SMA, PAMH1, HEND3, HEND3, TAC1. So if you cut again these plasmid with any of these restriction enzyme, and now you insert the gene of interest, the black one, you insert inside the LAGZ gene. Once this means that the LAGZ gene, which normally produce blue color when X-gal artificial lactose is introduced into the medium of the bacteria. But when it's interrupted with the gene of interest, here you can see that the colony will look like white. So once this gene is introduced, you can see that now the plasmid, after transformation and you grow up the colonies, you will see that these colonies will contain the recombinant plasmid. And here is this is looks like the shape of the plate on a medium with ampicillin and X-gal.